everything I've done, except my short stint at AMRI, had to do with proteins. And every time I read about one or learn or do any work with them, I just think that they're absolutely beautiful, amazing. They can provide structural support, catalyze reactions, protect the cell from foreign substances, and most interestingly to me is the new drugs that can be made from them, biotech. That's my background. That's really where I've come from. And when I first began my career, I studied the way small molecules interact with proteins. And then when I was in industry, I studied how proteins interacted with biological systems. So it's amazing. So first, we're going to spend time studying the building blocks of proteins, amino acids. We'll focus on their structure, and then we'll see how they behave at different pHs, titrations. And then we'll talk about, see how they interact, they react to form peptides. So again, the function of proteins is wide-ranging. They provide structure in tubulin, the one that I've talked about before, machinery, motor proteins, and they are information holders. They're organized. The information that they hold is in their three-dimensional shape. That's the information. So the computer system of our life is the shape of our proteins. So this is why I don't get worried about artificial intelligence, because they are just zeros and ones. That's the way that computers work. Until computers start to go three-dimensional, where the information is fitting two things together in a certain shape, then I will start worrying that they'll take us over and be like all those movies that we see. Now, we are going to study how they come together and form proteins. But some amino acids can be hormones and neurotransmitters. But their unique shape for proteins is really interesting. It can be very specific or selective or general. I have an example here. Glucokinase is an enzyme that processes glucose, but it can accept glucose, only glucose, as the reactant, the substrate, where another similar protein or enzyme, hexokinase, accepts glucose, galactose, or mannose, 3 different sugars. So you can have those differences between similar proteins. Both enzymes, the hexokinase and glucokinase, catalyze the reactions in the same way, but the difference is one is very selective where the other one is not. So as we saw in chapter 1, there are 20 naturally occurring amino acids that are linked to make proteins. The order of the amino acids is determined by our DNA, then gives the information to RNA in what process? DNA to RNA is transcription, and then the process by which the 20 amino acids come together, translation, right? Now, once we make that polypeptide or protein, we're going to bend it or fold it into an active 3-dimensional shape. So a protein won't do its job unless it has a specific 3-dimensional shape. And that's where we have that long rope of amino acids, that long polymer of amino acids folding in a certain way to give that 3-dimensional shape. And we're going to study those non-covalent forces, those intermolecular forces that cause the proteins to fold. Now, we can have really large proteins, which are always so hard to work with. Or you can have smaller proteins, which are much easier to work with. The large ones tend to have solubility problems, so when you're working with them on the bench, they don't dissolve, they just are not easy to work with. Dalton's is instead of grams per mole, we use the term Daltons. I don't know why. That's a good research topic. Why do we use Daltons instead of grams per mole for molecular weight and for proteins? Why don't we use barons? Joking, some dude's name. I don't know. And all right, so you'll find from textbook to textbook, there's different definitions between a peptide and a protein. For this class, we're going to use number of amino acids. So peptides are short chains of amino acids, and proteins are long chains. So we're going to define, just for this class, you might hear different definitions of 50 amino acids as a cutoff. Below 50, you have a peptide. Above 50, you have a protein. Now, some other textbooks, maybe this one, will define it as 5,000 Daltons. Below 5,000 Daltons, you're a peptide. Above 5,000 Daltons, you're a protein. All right, so let's start with the basics from the chapter 1 definition of the basic structure of an amino acid. Contains the amine group and the carboxyl group. And so the amino acids that make up our proteins are almost all alpha amino acids. And that alpha means there's one carbon in between the acid group and the amine group. Now that alpha carbon is very important. That alpha carbon has a hydrogen and then what we call an R group. That R group gives the identity to the amino acid. For glycine, that R group is going to be hydrogen. So here we can see this is the acid group, this is the amine group, this is the alpha carbon. The alpha carbon will have the hydrogen on it almost always, and then an R group. This R side chain is what's different between the amino acids. So you will see, and this is very important, this is the backbone of the amino acid. The acid, alpha carbon, nitrogen. So I expect you on the first exam to be able to draw the backbone of an amino acid. The general structure. I'm not going to have you memorize all of the amino acids yet, but what we're going to do is I want you to be able to draw that structure. So acid, alpha carbon, and amine. Alpha carbon has a hydrogen and the R group. Yes, go on.
Should we draw it with
No, you don't have to. You do not have to do a three-dimensional shape.
No, I meant like with hydrogen associated already.
Well, we're going to get to that. And maybe yes, maybe no. It's based on what pH it's at. If we look at the pKa, so what the question was, let me repeat that. Are you going to require when we draw the structure of our amino acid with it, the ionized carboxyl group and the protonated amine group? And it depends on the pH. So notice the pKa, this acid group will have a pKa. Same thing with the amine group. If you're above the pKa, the pH is above the pKa, this proton will be off. If you're below the pKa for this amine group, this proton will be on. At neutral pH, this is what the structure is. But we are going to go over this like I'm going to beat it into your head. All right? So this is why we went over titrations in the first place.
But for now, we just draw the whole thing like that.
Yeah, right now you can put the hydrogen on and hydrogen off if you want to. But before the test, we're going to go over what is what. So you'll learn, I don't know today, but at some point. All right. If the alpha carbon has 4 groups, so on that alpha carbon, you have the acid, the amine, the hydrogen, and the R group. There's 4 different groups on a hydrogen, or on a carbon. What does that mean? People that have taken organic chemistry. It's chiral. That means you can have a non-superimposable mirror image. So here we show alanine. Alanine has an R group that's CH3. So here, let's look at this. This is the alpha carbon right here. This is the alpha carbon right here. We have the acid group. So this is one group. We have the amine group right here. Hydrogen, and then the CH3. So on this carbon, we have 4 different groups. That means we have the possibility of having a mirror image that's non-superimposable. What does that mean? A mirror image that's non-superimposable. Your hands are not the same. If you have expensive gloves, not like the gloves that I have, you can only put the right hand glove on your right hand, your left hand glove on your left hand. The gloves that I own don't do that. Okay, I can put, okay, so I lose them. So I'm not going to spend that much money on a glove. But that means that they're specific, they're mirror images, and non-superimposable. So this sugar, glyceraldehyde, is the same way. Let's take a look at this one. This carbon, and when you draw it on the test, you don't need to do wedges and dashes. We're just trying to show you the three-dimensional shape here. Wedges come out at you, dashes go behind. But this carbon has a hydroxyl group, an aldehyde group, CH2OH, and a hydrogen. Those are 4 different groups. Now here we have the mirror image. So stereoisomers, that's the relationship to each other. They're stereoisomers, also called enantiomers. And we'll go over that again. But I want you, if you have a good imagination, pick this up, pick this right-hand one up in your mind, and try to place it on here, to superimpose it. What happens? Can you line everything up? No, what's opposite? Yeah, and how about, can you twist it to make it line up? How about flip this over like a pancake? What if you flip it over like a pancake? What happens? They don't face the right way. Okay, so it's not superimposable. It's because of that tetrahedral shape. So let's see. Say that my middle fingers are the OH, my pointers are the aldehyde, my elbows are the OH, and my thumbs are the hydrogens. I'm going to try to superimposable. So if I'm going to try to match. So, okay, this doesn't match up. And then if I do this, it's not going to, because of that tetrahedral shape, it's not going to line up, right? So that's why you have non-superimposable mirror images, same way as the left and the right hand. Now, we end up having an L and D designation, and it comes from our sugars. If you look on our sugars, like glyceraldehyde, you look at the carbon that is chiral. So we can say that this carbon is chiral because it has those 4 different groups on it. It's the origin of having a non-superimposable mirror image. So if you look, and if the sugar is over, putting the carbonyl to the top, if the sugar is on the right, we call it the D form. If the sugar is on the left, we call it the L form. And in sugars, the biologically active one is going to be the D form. But in amino acids, the biologically active one is the L form. Okay, the biologically active one. So if your body and nature only produces the L-alanine, because that's going to be the only one that is going to be biologically active. Can someone tell me what time it is? 9:30? Okay. So those are the L forms. Oh. Yes, for the sugars. Now for our amino acids, you can see that the R group is on the left. I am not going to ask you to identify the amino acids as D or L. I'm not going to have you draw me the L form of the amino acids, okay? Because then you'd have to do a 3-dimensional drawing, and I'm not going to do that. Yes?
Which one did you say was active for D? Was it sugar?
Sugar is active D, and amino acids. I'm not sure how loud this is going to be, but these are some great YouTube videos. What I do in my spare time.
So we're not going to have to be able to draw them? Are we just going to have to know how to,
A chiral object is
You're going to have to know.
like, be able to identify those now?
I want you to know from this what it means to have 4 different groups on a carbon, that you have a non-superimposable mirror image, that only one of them is biologically active. Okay? Okay, so let's go to my YouTube. Let's make it big.
A chiral object is one that is not superimposable on its mirror image. Your left hand is chiral. It cannot be superimposed on your right hand. Many molecules, including important drugs and natural products, are chiral. This is a ball and stick model of a carbon molecule with 4 different groups attached to the central carbon. For example, the 4 groups could be hydrogen, chlorine, methyl, and carboxylic acid. This molecule is chiral. That is, it is not superimposable on its mirror image. The 2 forms of a chiral molecule are called enantiomers. Using special rules, one form is labeled R, the other S. Enantiomers have mostly the same physical properties, but they behave differently in chiral environments. Molecules may contain more than one chiral center. For example, tartaric acid, which is found in many fruits and in wines, has 2 chiral centers.
And I always have to exit out really quick because very inappropriate YouTubes come up next. So you never know what you're going to get. We're not going to worry about the R and S designation. Those are mostly for organic chemistry. We're going to do the DNL here. But I hope you got a little bit better understanding there. All of the amino acids that we're going to study, except glycine, which has an R group that's hydrogen. So that means that there's 2 hydrogens on that alpha carbon, are chiral. Now there's the pair. They're called enantiomers. No, that's a spelling. There's an N. Enantiomers. Another thing is optical isomers. It's optical because they change the way that plain polarized light goes through them. That's how they discovered them. There's a lot of ways that we can call them. But we know that only the L form is going to be active for our amino acids. All right, now, because we're lazy and we don't want to draw out the full structure of amino acids, when we do the sequence of amino acids on our proteins, we usually give a 3 or 1 letter code, and you can find them in table 3.1. I just take a look at it to see how it looks. I'm not going to ask you, I'm not going to test you on that, but I think it's good for you to take a look at it. I know when I was in industry and we were working on a certain protein, I'd have above my desk the amino acid sequence. I remember when we would have different generations, I was particularly interested in the changes that they would make to the sequence of the amino acids and why. You know, to make it more soluble, to make it easier for it to get into the blood because you don't want to cross the PI. So there's, we'll talk about that later, but it's really very neat to take a look at why they would do that. So what I do want you to do is be able to take a look at the structure of an amino acid and classify it based on the R group, based on what's giving it the identity, as nonpolar, polar, acidic, or basic. So what we're going to do is go through those 20 and take a look at them. We're going to go through them in these classifications. And the reason why we classify them is because they behave chemically very similarly. So in our nonpolar side chains are these amino acids. We'll talk about them in a second. So I list them here. Let's take a look at these guys. So our neutral nonpolar ones are going to be glycine. That's the smallest one. Alanine is just a methyl group. So again, I'm not going to ask you, give me the structure of alanine. I'm only, the only one that's fair game now is the structure of glycine. So you can see that the backbone of the amino acid is everything that's not in blue. You have the amine group, the alpha carbon with the hydrogen, and the acid. That's what I mean by backbone. The stuff that's in the blue, that's the R group. Glycine's R group is just hydrogen. So that is fair game. I can ask you, draw the structure of glycine. Then what I can ask you is, I can show you another amino acid or just the R group of it and say, tell me if it's nonpolar, polar, acidic, or basic. So here, what do you see in the structure here, mostly, on all of these? What do we see? Just carbons and hydrogens, right? So if I show you one with just carbons and hydrogens, you're going to say neutral nonpolar. Now there's some exceptions. Tryptophan and phenylalanine, they have what we call an aromatic ring. And this is a benzene ring that you should have taken Gen Chem 2. You should have seen this benzene ring. It's very stable. And so here we have an indole ring, which is the two rings fused together. And and with a nitrogen here. So here, the nitrogen is not necessarily going in tryptophan, going to be accepting a proton because of the resonance here. Methionine, we do even though it has a sulfur, we do classify it as nonpolar because that sulfur, because it's buried, isn't really going to be doing much. However, you can draw an X over this cysteine. They do classify here as nonpolar, but I'm going to call it polar. I'm going to call it polar. And because it's tricky, I probably won't put it on that first test. Because some books classify it as nonpolar, and some books classify it as polar. I like to call it polar because it's involved in a lot of things here. And proline, look at that. It's just weird. Look at the R group. It comes down, and then it binds to the nitrogen. So it's a ring structure. What do you think that does to this bond right here? Is it going to be able to rotate freely? No. Now, I want you to remember the carbon, nitrogen, and carbon bond here in all of our backbones, there's free rotation. So what we end up with in our amino acid chain is sort of a zigzag. But is that going to happen here? No, not really, because there's restriction here. So we see this in bends and in very unique structures in our proteins. So when you see a proline, you take notice. You know, when I would look at that list and I would see a PRO, I would say, oh, there might be a bend. Or if I have an alpha helix, that might stop at that point when I see a proline. What's special about isoleucine in that R chain? Look at the carbons. There's nothing special about this guy. Nothing special about this. Yeah, that's a chiral carbon. Because if you look at this carbon, it has a methyl group, an ethyl group, a hydrogen, and then the backbone. So this one is chiral.
So we have to just know, we have to know all the names and then.
No, not yet. Later. For right now, I want to make it easy. You know, I did point out something special about proline. Okay, there's special about proline that I want you to know. And I want you to know that if I throw up carbons and hydrogens, it's going to be nonpolar. That's not too bad. Eventually, what we're going to do is we're going to be working with them. And the amino acids that we work with, you're definitely going to have to know those structures. So there's more interesting amino acids that that work in our active sites. I want you to know those structures and what they do. So as we work with them, we're going to learn the structure. Yes?
So even with proline and its ring structure, it's still nonpolar?
Yeah, it's still nonpolar because there's no carbon, it's only carbons and hydrogen, so it's nonpolar. It's just going to do some funky things to the structure. Yes?
What do you mean by work with them?
We're going to, we're not going to actually take them into the lab. What do we mean by work with them? We are going to see how they act in the active site of an enzyme, the mechanism of the reactions. We're going to see how some of them donate protons, hydrogen bond, and do the work in the active site. We're going to look at the chemistry of how an enzyme catalyzes a reaction. And I can't, do you think I get excited about things? I can barely contain myself when we do that, so just hold on. Okay, polar ones that aren't going to be ionized necessarily are these guys, serine, threonine, tyrosine, asparagine, and glutamine. what do you see in these structures? Either an OH, right, or an amide, right? So if you see an OH in the structure or an amide in the R, you're going to say that it's neutral polar. Again, the stuff that's in the blue is going to be the R part of our amino acid. That gives it the identity. So serine is just a CH2OH. We're going to talk about serine, a lot. The other ones we're not going to talk about too much, but we're going to talk about this a lot. So these necessarily, especially the ones with the OH. If you see an OH in it, it's going to be neutral polar. It's not going to be ionized. Acidic side chains are going to have acids, and then the basic ones are going to have nitrogens that are not an amide. Let's take a look at this. Aspartic acid and glutamic acid. Here we say 8 because we've already we've donated the proton, so this is the conjugate base. So I can show it to you, and I want to be clear about this. I can show it to you on the test with either the proton on or the proton off. So either or, you're going to classify this as an acidic side chain. So when you see an acid group, either it's in the ionized form or the protonated form, you're going to say that that's an acidic side chain. If you see nitrogen, as in lysine, arginine, or histidine, when it's not in an amide group, when you don't have the carbonyl next to it, so let me go back to this one. See here we have a nitrogen, but we have a carbonyl next to it. That makes it neutral polar. But here in our basic ones, you don't have that amide, the amide group. It's just the nitrogen groups on these. So in our lysine, especially lysine, we'll see that acting as a base. Arginine and histidine can do that as well. Histidine, actually, we're going to work with in our example of the chemistry at an active site. So remember, a base can accept a proton. Here, I'm showing it in a protonated form, where it has accepted the proton. Again, I can show you to you as the NH2, where it hasn't accepted the proton, or the NH3, where it has. Okay, it just depends on the pH. So the difference between aspartic acid and glutamic acid is that the length of the chain before we get to the acid. Lysine's pretty neat. I like it. It has a really long chain before we get to the base, so it can reach out pretty far. I like that. Arginine can do that too before we get to the guanidine. This is a guanidine actually group there. And then histidine is very stabilized. When it has that hydrogen on there, that hydrogen ion on there, it's stabilized within the ring. So that's why it acts as a base. Any questions? I think I've made some generalities for you to remember them. Yes?
Is it worth just writing down the R groups? Like, I know we don't know the structure right
Yeah, so again, the question was, is it worth knowing all the R groups? Remember, this class is a 200 level class, and some of you would need this for, MCATs, and some of you just need it because you just want to take it because you're just a cool dude. And so it depends what you want out of this class. If you're taking this for the MCATs or to get into another program, yeah, you should probably start memorizing them. I do not require that you remember these, but I just want to point out that there are a lot of derivatives of amino acids. In groups or in areas where we need a lot of hydration, you'll see hydroxyproline. We add groups to these other things. So hydroxylysine and hydroxyproline we'll see in connective tissues such as collagen, and what that hydroxy group is, it draws water to it. Thyroxine is only found in the thyroid gland. Remember, you need the iodine from the salt, so we use the iodized salt so that we can produce our thyroxine for our thyroid gland. Neurotransmitters can be derivatives of glycine. A glutamine derivative is serotonin. and a tryptophan derivative is serotonin. And melatonin is also a tryptophan derivative. My husband and I take this at night to help us sleep. But if you take too much, it's like you have a hangover the next day. You want to just sleep all day. So be very careful if you take that. Does anybody take melatonin? You have before? Does it help you guys?
No.
Sleep?
I've never experienced anything different.
Yeah. It makes me like sleep hard. Yeah, it makes me a zombie. You know what helps me better is Benadryl. If you can't fall asleep. Yeah, when you get to be elderly like me, sleep comes easy, but not, you can't stay asleep. So you just take a little bit of children's Benadryl and you're out. So again, thyroxine, and indole acetic acid, they are found in the thyroid gland.